Hello students, welcome back to the biology class. So in the last two classes we started the concept of genetic material, and we understood that how the properties of genetic material, or what criterias were given by the scientist to call some biomolecule as a genetic material. And we learned also that what facts theoretically we have that proves that DNA is the genetic material, and then how experimentally Griffith performed the experiment with the help of Diplococcus pneumonia or Streptococcus pneumonia, which has two strains. One is the S strain and the other is the R strain. These experiments slowly gave rise to the clear understanding to the worldwide that nucleic acid is the genetic material, and then it became clear lately that RNA came first, DNA came later, but DNA is a better candidate as a genetic material compared to the RNA. Genetic material is a medium through which the information passes from generation to generation, and it also controls the formation and expression of the traits in the organism. The various properties we learned was that amount should remain same in all the cells, should be structurally and chemically stable, should be able to create its own replica formation and then pass on the copies in the same manner to the next generation, should allow some amount of adaptation, some amount of changes, which is called as mutation, which is helpful in adaptation as well as evolution, and express in the form of Mendelian character. Then experiment started, and then Griffith performed the experiment with the mice and the Diplococcus pneumonia, which had the two strains. We learned that this experiment made it clear to him that there is something, but he never understood what was that biomolecule. But he stated that hypothetically there is a biomolecule which he could not extract or isolate because of the problems with the technology at his time. In 1928 he performed the experiment. So he only stated that the presence of the heat-killed S strain bacteria has caused the transformation of the R strain into the S strain. So whenever a bacterial cell divide by binary fission or amitosis, so R strain, instead of giving rise to the R strain two daughter cells, gave rise to S strain. So this happened because it picked up the DNA from the surrounding, because in the surrounding there was the genetic material of heat-killed S strain bacteria. So this was confirmed that the transforming principle or transforming molecule is DNA by Avery, MacLeod and McCarthy, because they performed experiment in two methods. One was dividing the filtrate into four components: protein, lipids, RNA and DNA, and they found only whenever the DNA of the heat-killed S strain bacteria along with the living R strain bacteria were injected into mice, then only transformation occurred, or you can say then only the mice died with infection. And when they performed autopsy or post-mortem of the dead mice, they found living S strain inside their body or in the blood. The second setup of experiment they did was they divided the filtrate into four parts and treated these parts with various enzymes, and they found that only when DNase is used, then only transformation did not occur. That means DNase has digested DNA, that's why no transformation occurred. This confirmed DNA is the transforming principle. Then we learned regarding the Alfred Hershey and Martha Chase 1952 experiment, in which I gave you the background how they developed radio-labeled virus. And then after developing these radio-labeled virus, they infected the radio-labeled viruses to the normal E. coli. So the first step was infection according to the NCERT, because NCERT does not give you the background, it only starts from here. So infection, blending and centrifugation and detection of the radiations are the four steps. NCERT call it as three steps, as infection, blending and centrifugation. Once infection was done with the virus, one virus has the radio-labeled DNA with phosphorus 32 radiations coming out from it, and the other virus had its protein coat which is radio-labeled with sulfur 35 type. When blending was done, after infection, the leftover viral portion was separated from the bacteria. Bacteria is heavier in size, remember. So when centrifugation occurred, the pellet separated from the supernatant, and then they discovered that the radiations of sulfur 35 was coming from the overfloating portion, supernatant portion, and they discovered that this phosphorus 32 radiations were coming from the inside the bacteria, which was the pellet. So this confirmed that DNA enters inside, protein does not enter inside. That's why radiation of only phosphorus 32 came from this experiment setup, and in this setup there is no radiation of sulfur 35 from inside the bacteria. That means protein does not enter inside, but radiations were coming from the supernatant. So this confirmed that proteins are not the genetic material, but DNA is the genetic material. So this confirmed that DNA is the genetic material, and now universally everybody started trusting this concept. And then comes the topic of the replication of DNA. Before we start with the replication, let's understand the background regarding how replication became clear to scientists. So Watson and Crick were the two scientists. Remember, these were the one who gave the double helical model also. They proposed that DNA replication is semi-conservative. Now let's understand today what exactly is semi-conservative. It's a type of replication in which one strand of daughter duplex means that double helix, is derived from the parent while the other one is formed anew. The two strands of DNA molecules separate and each strand serve as a template. Template means a guidance. Like for example, if one strand is written as C, then obviously the other strand will have G because of complementary, which you have learned already in 11th as well as 12th class. So when you have the reading of one strand, you can easily predict the reading of the other strand. So that's why the one which is given to you is called as the template strand. For the formation of new strand, that new strand would be called as the complementary strand. Thus the new molecule of DNA would be made up of one old strand and the other would be the